Hello everyone, today I'm happy to provide you with the overview of the AnnotSV workflow. First, I would like to explain the context of structural variations that I'm going to call SV, so to explain the context of SV in human genetics. SV are generally defined as variation in a DNA region that vary in length from 50 base pairs to many megabases, and SV includes several classes such as duplications and deletion, and also translocation, inversions, breakpoints, and so on. Human geneticists generate large amounts of genomic data, including millions of small variants, but also thousands of SV, mainly from array-based techniques and next-generation sequencing. Relevant annotation are available from many well-known databases like OMIM, DGV, ClinGen, NOMAD, and some joint consensus recommendations of ACMG and ClinGen was published in 2020 for the interpretation and reporting of SV. But the SV annotations ranking and analysis are still challenging. Geneticists have to filter out the SV to identify the one pathogenic variation among thousands of variants that is responsible for the patient's phenotype, which is a bit like searching for a needle in a haystack. That's why, to help identifying human pathogenic SV, we have developed a software, AnnotSV, dedicated to their annotation and ranking, as well as a web server, not AnnotSV, dedicated to their visualization and interpretation. Okay, so here is the overview of the AnnotSV workflow. First, the user needs to submit the SV genomic coordinates from a single patient or from multiple patients. In this way, AnnotSV provides to the user two different input file formats, a BED or VCF file, and AnnotSV is compliant with all types of SV. Many options can be set in input, as the genome build, for example, that is very important. And moreover, something that is really interesting, AnnotSV allows the user to do a phenotype-driven SV analysis. In this aim, the user can also provide the phenotype of the patient as a list of HPO terms, which are a standardized phenotype codification. So quickly, for those who are not familiar with HPO, you can get them from several websites like hpo.jax.org, and you simply enter a phenotype name and you get the corresponding HPO term, as you can see here. Phenotype is optional, but it's highly recommended to use it for better prioritization. This means that the user can identify relevant SV based on how similar the phenotype of the patient is to the gene overlapped with this SV. Okay, so once the user has submitted these input data, AnnotSV integrates all of these in the first step. Then AnnotSV annotates the SV with more than 60 sources, representing more than 100 fields in the output, and that allows the user to explore different informations. AnnotSV reports the genes overlapped with the SV, as well as gene-associated annotations like protein, gene intolerance, haploinsufficiency, and so on. And AnnotSV also aggregates general population frequencies, non-pathogenic regions or regulatory elements overlapped, and breakpoint information. Moreover, AnnotSV permits some custom annotations to be added. For example, regions of homozygosity for a patient or internal lab frequencies. Then, if the user has provided some phenotypes in input, the Exomizer module scores each overlap gene depending on its phenotype. Basically, the more the gene matches the patient's phenotype, the higher the score is. This score is between 0 and 1 and becomes very specific when it is greater than 0.7. Then, AnnotSV apply STMG CleanGen rules to score and classify SV among the 5 classes from benign to pathogenic. And finally, there are 2 kinds of outputs. A tab-separated file, as you can see, produced by AnnotSV, that you can integrate directly in your personal bioinformatic pipeline, and an HTML file produced by not AnnotSV that you can visualize directly. It's a ready-to-use display for SV analysis and interpretation. Well, that's it. Feel free to look at another tutorial video. Bye-bye!